In this video, we're going to go over how to analyze different human pedigrees. So there are 3 steps to ask yourself as you look at a pedigree. First of all, are there any affected family members in every generation, which we would call a vertical pattern, or is there only single generations affected? So do you see a horizontal pattern? The next question, once you establish the mode of transmission, then you're going to ask yourself, what is the ratio of affected males to females? So look for differences in the genders of those that are affected. And then the last step and last question to ask yourself is the disease or gene being passed through unaffected females. So is there a male-to-male transmission also? And what percentage of the children are affected? For example, are all the sons but none of the daughters? So you're going to look for, again, some sort of gender transmission that's occurring and patterns in that transmission. So with these types of questions, you can determine whether what is being passed is dominant or recessive. And then with these questions 2 and 3, you can figure out if it is a sex-linked or autosomal trait. So let's take a look at that. So here is a sample pedigree of cystic fibrosis. And when you look at this, you see the affected individuals are the darkened square and circle. The squares are males, the circles are females, and we can see that's even labeled right here. And we have the I, and then the double I, and then the triple I here to represent Roman numerals 1, 2, and 3, showing the 3 generations. Each generation is in a horizontal line. And you can see these are the parents and their offspring are connected directly to this offspring line. So they have 1 daughter, 1 son, and then a second daughter. And then the lines that are parallel here and horizontal show, again, a couple. And here this daughter has married this man. And then this line will show now the offspring from this pairing. And here we have 3 daughters and 1 son. We have 1 affected daughters. So asking those questions here, what's the mode of inheritance? So do we see this occurring in every single generation? So we actually don't see it in every generation. What we do see it in 2 generations, but we don't see it here. So what we're trying to determine is this dominant or recessive? And in looking at this, you wouldn't say that it's dominant, because if it were, then you would see a lot more occurrence of it. And if it were dominant, in order to have an affected offspring here, you would have to have at least one of these parents having the disorder, because that means they had to have a dominant allele to pass down, and they don't. And so you cannot have unaffected parents have an affected offspring if it's a dominant condition. So in this case, we would say it's recessive. So now the next question is, are the genders affected individuals equally represented, both males and females? And in this case, yes, they are. And so when they're equal, we're going to say it's not sex-linked. It's going to be something that is found on an autosome. And it is true that in cystic fibrosis, it is an autosomal recessive disorder. So if it were autosomal dominant, this is what the pattern might look like. So in this case, what we're seeing are multiple generations affected. And so now we see at least every generation has at least 1 person affected in it. We call this a vertical pattern. We also are finding that males and females are equally affected. So we've got 2 females and 3 males. So it's roughly the same. And so you would say that it looks as though it's not sex-linked. It seems to be autosomal. But we can ask some more questions now. So do we see male-to-male transmission? And so here we have male and then male. So we do see some male-to-male, but it's if it were male-to-male transmission, it would be a Y-linked condition, and we have affected females. So that's not going to be the case here. So when we're saying male-to-male transmission, we mean only male-to-male transmission. We see each child of an affected individual has a 50% chance to be affected. So we look at each affected individual, and this couple or this individual that's affected has 4 children, and half of them have the condition. In this couple here, this affected individual, half of the offspring have it. And here we have 1/3, but that's close enough for an autosomal dominant. You can also see that unaffected individuals do not pass on the gene. So if you have unaffected individuals, here's an unaffected individual whose spouse is also unaffected, you will not see any transmission of the disease to their offspring. And then finally, to be autosomal dominant, every affected child has an affected parent. So you have to look and see that type of inheritance pattern occurring. So this affected child has an affected parent. This affected child has an affected parent. And you can trace that all the way back. Back to the male-to-male transmission. The other thing that's showing us, there is male-to-male transmission. So what that is showing us is that it is not on the X chromosome as well. Okay, so we looked at this one already. Okay, so this is an example of an autosomal dominant pedigree. So taking a look at this one here, the purple are the affected individuals. So we see a female here who actually had 2 male partners with 2 different sets of families, essentially, 2 sets of offspring. And so in both cases, we look at the transmission rate and we're seeing this generation has it, this generation has it, this one does, and this one does. So we see vertical inheritance here. So now we're going to take a look at, do we see equal representation in males and females? And if you were to count this all up, it looks like it's pretty evenly divided. So there's 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, 24, 25 females. And let's see, 2, 4, 6, 8, 10, 12, and 14 males. So again, it's not exactly 50-50, but there are enough of both genders to say it's not on a sex chromosome. And then you want to check finally, does every affected individual have an affected parent? And so you can trace backwards and see that yes, indeed, that is the case. And here's affected, affected, these both have affected. Okay, so the other thing that you should see is that you see a lot of incidents, a lot of affected individuals in this pedigree. So that's another clue. When you see this many affected individuals, you're going to immediately suspect that it's autosomal dominant. So an example of an autosomal dominant trait is dwarfism. So achondroplasia is another is the more scientific word for dwarfism, and it is an autosomal dominant human disease. So we don't see a lot of autosomal dominant diseases because when they are dominant, again, you see a lot of affected individuals. And if they are harmful to the lifespan or longevity of an individual, then they're really not going to live long enough to have offspring and have the future generations carry that disorder. But in the case of a dwarfism, for example, it's not life-threatening as a condition. People can live a normal, long life with this condition, and so we do see incidences of this in the human population. So this is a pedigree showing achondroplasia, and you can see here we see vertical transmission. Every generation has it. If we count the males, there's 2, 4, 5, and there's 4 females. So you see it's autosomal. And then every affected child does have an affected parent. Okay, so now let's take a look at autosomal recessive. So in autosomal recessive conditions, you are not seeing as many affected individuals. So that's key. I want to point out something in this pedigree here. You see these dots in the some of these individuals, circles and squares here. And what that's showing, if you see something like that, most likely what they're trying to show is a carrier. I'm going to tell you in AP Biology, we don't usually show the carriers. So these would be blank in most of the problems that you're going to be seeing. But just maybe for learning purposes, now you can kind of see where the carriers are. So things to look for when it's autosomal recessive. So the first thing, let's check to see, is it dominant or recessive? So we're going to look to see, is it in every generation? And we only have one generation here that has affected individuals, and we have 2 that do not have affected individuals. So right away we're seeing we have horizontal inheritance. There's only one horizontal generation where we're even seeing any affected individuals. So that's one telltale sign that it is a recessive condition. Now the next question is, are males and females equally affected? And in this case, yes, they are. So again, that will tell us that it is on an autosome and not on a sex chromosome, an X or a Y. Other things you can notice and that need to be seen in an autosomal recessive pedigree is that the parents of affected children can be unaffected gene carriers and have a 1 in 4 or a 25% recurrence risk. You could also say this, a 25% penetrance. So penetrance, genetic penetrance is the actual what's the right word to say? It's the actual occurrence of the condition. So you actually see the phenotypic trait. You can see that unaffected siblings have a 2 out of 3 or a 67% chance of being carriers, and children of affected individuals are obligate carriers. So what that means, they have to be a carrier because in order to have the condition, you need 2 recessive alleles. So that means if you have a parent who has a condition, then all of the offspring will be carriers, depending on what this other parent. They will have at least one of these alleles. And you'll see that here and here. The other thing is if you have 2 unaffected parents and you do have children that have the condition, then you know that those unaffected parents have to be carriers. They have to be heterozygous as well. So this is a much more common sort of pedigree that we'll run into. So that you'll see again, the trait is rare in the pedigree. It often skips generations because it's hidden in the heterozygous carriers, and it affects males and females equally. So here are some of the common types of autosomal recessive diseases in humans. So we see cystic fibrosis, sickle cell anemia, phenylketonuria, or PKU, and Tay-Sachs disease. So these are all common autosomal recessive disorders. For each of these, overdominance, which is heterozygote superiority. We also call it heterozygote advantage, has been suggested as a factor in maintaining the disease alleles at a high frequency in some populations. So what does all that mean? So what that means is when we have heterozygotes, in some cases, there's some evidence that people who are heterozygotes actually have some sort of health advantage in the environment in which they live. So in this case, we see this in sickle cell anemia in particular. We know that this is the case where individuals do have a heterozygote advantage. So what that means, if you are homozygous recessive for sickle cell anemia, you're pretty sick. It's a pretty debilitating disorder. But if you're heterozygous for sickle cell anemia, meaning you only have one allele of the recessive mutated allele for the disease, then you are not as sick. You can function, and you're not 100% healthy, but you can definitely function better than someone with the autosomal recessive condition. So people who are carriers or who have the heterozygous condition, what we have found and discovered is that they do not get malaria. So in areas where malaria is a threat to a person's health, and in many cases it's even more debilitating than the sickle cell anemia, then being heterozygous actually gives you a health advantage over people who don't have any of these alleles. So if someone is autosomal dominant, they're much more likely to get malaria than someone who is heterozygous or actually has sickle cell anemia. They're homozygous recessive. So that's the heterozygote advantage superiority, or another word for it is overdominance. Okay, so let's take a look at the next type of inheritance pattern here. So in this case, we're going to be looking at X-linked recessive pedigrees. So in this case, we see the trait is rare in the pedigree. It's skipping generations. So that tells us it looks like it's a recessive inheritance pattern. But now when we get to our second set of questions on looking at which gender is being affected, we do not see an equal amount of males and females with this condition. In this case, we're seeing only males being affected. So when that's the case, we are going to suspect that it's X-linked in addition to being recessive. So it doesn't look like it's on the autosomes. Now, we know it's sex-linked when we're only seeing one gender affected, but we don't necessarily know it's on the X versus the Y chromosome. So the way that we're going to determine that is by looking at how fathers and sons relate to each other. So when we see affected fathers not passing it on to their sons, then it is on the X chromosome. So males are going to be more often affected than females if it's an X-linked recessive condition, but females can get it. So females who are homozygous recessive, both of their X's have the allele, then they'll be affected as well. So you will see a few females affected, but males are much more likely to be affected. So here's an example. This is a famous example of an X-linked recessive trait. So hemophilia is one of the conditions that is X-linked and recessive. And you can see in this particular case, we do see it skipping generations. So these generations don't have it, although they're showing you she is a carrier. And then here we have another woman who is a carrier. So when it's X-linked and recessive, of course, you'll never see a male carrier because they only have one X chromosome. So if that X is affected, they are affected. So in the case here, this individual's X chromosome has that allele. And so then we look to see, well, this person didn't actually have children. Let's see if we can find any males who did. We actually don't have an example of any males who did have children. So we can't look at that in this particular pedigree. We are seeing more of it than we would expect. So we do see it in these two generations. And this particular case is different because, and the reason you're seeing more of this than you typically would with a recessive disorder is because European royalty were infamous for what we would term inbreeding. So they would marry and have children with their cousins. And so when that was the case, when you have an autosomal or an X-linked recessive trait, and you have close relatives marrying and having children, then that recessive trait is going to show up a lot more. And so that's what we actually see occurring here. I'm trying to see, where's that example? Well, here's an example right here. So these two are related because this person is related here and this person is related over here. So these are first cousins. And you're going to see this in other places too. If you look at this carefully, you can find those. You can pause them and find more examples of that. Okay, so what about X-linked dominant conditions? So X-linked dominant conditions are going to be rare. Females are about 2 times as likely to be affected than males. And oftentimes for males, it can be lethal. In the females, it can be milder. Affected males pass the gene to all of their daughters who will be affected and to none of their sons. So you see no male-to-male transmission, and sons and daughters of affected females have a 50% chance of being affected. So it's similar to an autosomal dominant condition. So in this case, you're going to count up here and see there's 1, 2, 3, 4, 5, 6, 7, 8 females and 1, 2, 3, 4 males. So again, about double the females to the males are affected here. You see it in every generation. So that tells us it's dominant. And so now let's take a look at the transmission. So if we have an affected male, we want to take a look at the affected males and see if they pass it on to their sons. So we do not see that, but all of their daughters do have it. So if we look here, here's another male, all of the daughters have it. Okay, and here again, here's an affected male, all of the daughters have it. So this is important in determining that it is X-linked and dominant, and it's not just a dominant condition. Let's see. Okay, so I think that is pretty clear. So here's another example of an X-linked dominant pedigree. So again, it's common in the pedigree. You see a lot of it. Affected fathers pass it to all of their daughters, And males and females, it says here, are equally likely to be affected, but you're going to actually see it more in females. So let's see here. 1, 2, 3, 4, 5, 6, 7, 8, 9 females, if I counted right. And 1, 2, 3, 4 males. So females are about twice as likely to have it. And again, that's because they have twice the number of X chromosomes. So that would make sense that they're twice as likely to get it. If you look at the fathers, you can see all of their daughters have the condition. So here's an affected male here again, and all of the daughters. Here's an affected male, all of the daughters, affected male, all of the daughters, and none of the sons. So some X-linked dominant diseases. So rickets is a bone lesion disease, and this pigmenty is a skin lesion disease. So these are two, and again, they tend to be rare conditions because often they're lethal to males even before they're born. Okay, so what about a Y-linked? A Y-linked condition is also called a Holondric condition. So in this case, of course, only males have a Y chromosome, so only males will be affected. So how do I know this is not, if only males are affected, I would think, okay, well, is it X-linked? And so in this case, the way that we know that it's not X-linked is then in X-linked, fathers never pass it to their sons if it's X-linked. But in this condition, all sons get it from their fathers. So every affected father gives it to his son. So you'll see this here. And you again see it in no females. And it's not dominant or recessive because there's only Y chromosome. There's only one Y chromosome. So it is dominant essentially. So what I'd like you to do is for each of these pedigrees, identify the mode of inheritance. So is it dominant or is it recessive? And then tell me, is it autosomal or on a sex chromosome, an X or a Y? And give 2 reasons for your choice. So pause this and think about it. Think of your reasons for why you're choosing what you are. Okay, so what you should have come, now hopefully you have figured this out before continuing. So what you should have seen here is that, of course, it is not in every generation, so it looks like it's recessive. And then we see affected individuals without affected parents, which tells us, yes, it is a recessive disorder. And then we also see males and females equally affected. So we say this is an autosomal condition, so it's not on a sex chromosome, and it is a recessive condition. Okay, so this would be another example of that type of inheritance pattern, where we're seeing, of course, very few incidences or occurrences of the disorder. We don't see males and females, but we do see females who do not have, for example, a male passing it down, so it's not something that is on the X chromosome, for example. It could be X-linked recessive because we really don't know what this female, I mean, she could be a carrier, but since we are not seeing any males at all, then we would really lean towards it being an autosomal condition, not an X-linked condition. Okay, so what about this one here? So pause this. determine what type of inheritance this is and give 2 reasons why. So what you should see, hopefully you've paused and now you're resuming. So what you should be seeing here is something that's skipping generations. So it looks like a recessive disorder. And what we're seeing also is only males being affected. So immediately we think this is X-linked. It could be X or it could be Y. It's going to be sex-linked for sure. So if it were Y-linked, then every affected father would have affected sons, and we're not seeing that. So that means this is an X-linked condition. So you can test yourself and make sure that makes sense. So if that were the case, and this affected individual has an X chromosome with the disorder, is it possible for him to have daughters that don't have the disorder? And yes, with an unaffected mom, then that means that this mom has to be homozygous recessive. I mean, homozygous dominant, oops, because she has to be giving the dominant allele to cover up the recessive X allele here in order for these daughters to not have the condition. And then here, this affected individual, is it possible for this individual to be affected with these parents? Well, most certainly, every one of these daughters is a carrier. So they're all heterozygous. And if they're carriers, that means that the X that they're giving their offspring is either going to be 50% affected and 50% unaffected. So the chances that this male is going to get that affected X, it has a 50% chance of happening. So it can happen. It is definitely plausible with this type of transmission. So based on that information, we would say this is an X-linked recessive disorder. Okay, so now what about this one? So pause and analyze this one. Okay, so what we're seeing here is every generation is affected. So immediately we're going to think, well, it doesn't look like it is a dominant condition, a recessive condition, because we do see vertical transmission occurring here. So now let's look at males and females. So in this case, we have 1, 2, 3, 4, 5, 6, 7 males and 1, 2, 3, 4 females. So we're seeing, even though, again, those are not equal numbers, we would still say it's occurring enough in the females to suspect that it's not on the X chromosome. Is there anything else that would tell us it's not on the X chromosome? Well, right here, this definitely would tell us that it doesn't seem to be on the X chromosome. Because if it's a dominant condition that we're seeing in every generation, then that means that this male could not have gotten it from the dad. It would have to have come from the mom. And so if she had one dominant allele, then that would make her affected as well. So that doesn't work. So we suspect that it is not on the X chromosome. I would say that this looks like it is autosomal dominant. So again, the penetrance is vertical. Each generation has the disease, and every individual who is affected has an affected parent. Okay, so I want you to pause and figure this one out here. So the first thing you should have noticed is that this generation, well, actually, this is part of this generation here. So we are seeing it in every generation, and every affected individual does have an affected parent. Except, well, let's look right here. This affected individual does not have affected parents. So maybe not dominant, even though it's in every generation. So now, because of this, we cannot say it's dominant. So it must be a recessive condition. So now let's look at the males and females. So we have 2 males and 3 females. So it looks again like it's not sex-linked. So this is an autosomal recessive disorder. So now let's figure out the genotypes based on that. So it's autosomal recessive, meaning if you are affected, you are homozygous recessive. So this individual is homozygous recessive, and so is this one, so is this one, and so is that one. And so is this one over here. But now let's figure out, okay, so this one we know is homozygous recessive. What about this one? I mean, this one over here. So if this one's homozygous recessive, this individual, that means this person is going to give one affected allele. And so they did here. Each of their offspring has to have at least one affected allele. Now, in order for the children to end up being homozygous recessive here, that means this individual who is not affected has to be a carrier in order to give them that a recessive allele as well. So this person is heterozygous. Again, the offspring are homozygous recessive. This person is unaffected, but they had to get the recessive allele from the mother here. So this daughter is also a carrier. So if she's a carrier and her spouse is from this family here in this generation, and actually this is taking an autosomal recessive condition and having cousins again, producing offspring, then we've got more likelihood of having the disease show up here. But this individual here, so we know this person's a carrier. This person is also a carrier, which is why we end up with an individual here who is autosomal recessive. So this person could be a few different things. So this person could, if this person is a carrier and this person is a carrier, then the unaffected is either homozygous dominant or they're heterozygous. We really don't know which one that would be. So this is the answers that should have come up with. Okay, so this is another one. I'd like you to pause and figure out the genotypes. Okay, so you've resumed now. So let's go through this. So this is an autosomal dominant condition. We see it in every generation. We don't see it here, but we do see, well, actually we do see it in this generation. It's over here. So it is in every generation. So it has vertical transmission. It's dominant. It's autosomal because we see it equally represented in the males and females, at least equally enough. And we do not see it only occurring in the males. We see quite a few females, actually. quite a few more. So it's not X-linked. And it's not Y-linked because not all sons are getting it from affected fathers. So it definitely is on an autosome. That should have an O there. So let's take a look now at what type of genotypes we have. So the first one here, since it's dominant, this individual has to be heterozygous because if it's a dominant condition, if they were homozygous dominant, then every one of their children would be affected because they would all get the dominant allele. So we know that this person has to be capable of giving the recessive allele in order to have normal children. And the same thing we see here with B, because this individual has one normal child, which is not possible, especially when we've got two parents that are affected. So we also know this mother here has to also be heterozygous in order to produce an unaffected child here, a son and a daughter that are unaffected. C, in order to be unaffected, all of the unaffected individuals are homozygous recessive. And so that gives us the answer for D as well. For E, we don't know. We would have to say that this individual is either homozygous dominant because it could have, she could have gotten the dominant allele from both parents, or she could have gotten the dominant allele from one of her parents and the recessive allele from the other of the parents, because we've established that these are heterozygous parents. So this genotype would be either one of those two things. So let me go back here. I totally lost my slides. Okay, so I wanted to end with these slides here. So these are some of the things, options that you have when you're looking at patterns of genetic inheritance. So for Mendelian inheritance patterns, again, it's either going to be autosomal dominant or autosomal recessive, and it's either going to be sex-linked. It would be X-linked recessive or X-linked dominant. And again, remember, those are very rare. Or Y-linked, which is also called Halandric. We also have non-Mendelian patterns of inheritance. So occasionally you're going to see mitochondrial or non-nuclear inheritance. And that I'm going to show you in a minute how to identify when that is occurring. Some of the things you won't be able to really tell from a pedigree, and actually would not be in a test question at this point, but these are just things to be aware of. So things like imprinting or multifactorial inheritance can mess up a pedigree. So when it's imprinting, that is something we're going to actually learn in the next unit and talk about when we talk about epigenetics. But imprinting is an epigenetic process that requires the methylation of DNA and the methylation of histones, and it does not actually change what allele a person has. But the epigenetic markers are imprinted. So this methylation sequence, so when DNA is methylated, it is silenced essentially. And these markers are imprinted in the sperm or egg cells of parents. And so during meiosis, this is established, and they actually believe it occurs after meiosis has occurred, but it's maintained through the mitotic cell divisions in the somatic cells of an organism. So once fertilization has occurred and mitosis begins dividing and copying the fertilized zygote, then that imprinting continues throughout the person's lifetime, we believe. So this is actually not entirely verified, but this is what we believe occurs. So essentially it means some alleles get silenced through imprinting. So you may not see the true autosomal dominant or recessive pattern showing up. Multifactorial is when a combination of genes from both parents plus unknown environmental factors make the trait or condition appear. So we don't see the phenotype unless we have the correct combination of genes from both parents, plus environmental factors is another way of saying epigenetic factors as well. These are also some more complicating factors. So penetrance is, again, if a person has the alleles for the condition, let's say it's a autosomal dominant condition, yet they do not have the phenotype, they don't show the disorder, then that means they have non-penetrance of that gene or of that condition. So the individual has a trait-associated or disease-causing genetic variant, which is another way of saying an allele, but the individual does not have the phenotype or the condition. So that's non-penetrance. Sometimes there are new mutations, so that maybe there's a completely new allele that has shown up. So that you would not be able to figure out in your pedigree. Adult onset conditions. So maybe the pedigree information, which is again being obtained just based on whether the person has the condition or not. Well, sometimes if you do it before the person has actually developed the condition. So for example, with Huntington's disease, it doesn't show up until a person's in their 20s generally. And so if you've done your pedigree analysis too early, you wouldn't actually see the true incidence in the family in generations. Consanguinity is the degree of relatedness between individuals. So this is going to be a factor that can sometimes cause the genes that they're sharing between them, so the two different alleles, to be showing differently. So we saw this example when we looked at the royal family, the European royalty, and how they showed a recessive disorder looking a lot more like a dominant disorder because of the fact that too many closely related people were having children. Sex-limited or sex-influence conditions, sometimes just, for example, there are other factors based on gender, such as hormones, that might affect the expression of a gene. Germline mosaicism, that's a hard word to say, is when a sperm or an egg can have a mutation that's not in other tissues of the body. So this is when you have mosaicism, means that you have not the same gene expression occurring, or the same genes actually being, well, I guess expressed in different parts of the body. And pleiotropy we already talked about. So let me pause this for a second. Oh, it's not going to let me pause it. Okay, so the last inheritance pattern I want to just show you is mitochondrial or non-nuclear DNA inheritance. So let's take a look at this. So what we know about mitochondrial inheritance is that you only get your mitochondria from your mother. And so it's only passed down through females. So affected females will have all of their children will have that condition because, again, mitochondrial DNA looks different. Remember, it looks like prokaryotic DNA. So it's not going to have, two different chromosomes. It actually has one large ring of DNA. So it's going to have a little bit different look to it. So the way that you tell it's mitochondrial inheritance is you look at affected females and you see if all of their children are affected. And then the other thing that you will see is that if you look at affected males, none of their children have it. So you can take a look at this pedigree and see that that is definitely the case. And whenever you see this pattern, then you have to assume that it's mitochondrial, or what we'd call non-nuclear DNA inheritance. Okay, so what I'd like you guys to do now is, again, go through this again if you have questions. There is a video for you to watch on Google Classroom on a pedigree analysis. So I'd like you to watch one more video and then do some practice problems. So I want you to really get comfortable and check your answers. There's also going to be an answer key for the not mitochondrial, for the pedigree practice problems, so that you can make sure that you're getting it correct, so that by the end of this class period, you are comfortable and confident in your ability to analyze these different pedigrees. Then if you do still have some time left over, please just work on any number of activities. You can work on your dynamic study module for Chapter 15. You can work on Chapter 15 reading notes. There's plenty to do to fill up your time today. Okay? And if you have questions, please put them in put them in an email to me today, or you can place them in the chat before you leave. If you've signed off, then just put them in an email, and then I will address them in our next class period. Okay.